Welcome back, folks. Today, in this video, we are going to talk about enzymes. The next lesson in digestion, which is really important, is going to look at some specific digestive enzymes and how they break down polymers into monomers. But what I want to do is just take a couple of minutes here. This is a nice short presentation, but you do need to know this one. What are enzymes? And how do they work? And how do we sort of stop them if we need to sometimes? How can certain things stop them sometimes when we don't want them to? But remember that enzymes are proteins and they speed up chemical reactions. And I would guess that 99.99% of chemical reactions in your body require some help to go as quickly as they do. They require an enzyme. So the only reason you and I are alive, the only reason that I'm talking right now, and the only reason that you're listening right now is because of enzymes. So they're very, very important to life for humans. And also life for all organisms. I should throw that in there. Just to quickly start metabolism. You've probably heard about that term or heard that term before. We talk about some people having fast metabolism, some people having slower metabolism. Metabolism is actually, in a biological sense, just all of the chemical reactions in a cell. Some of those chemical reactions are putting things together. Some of those chemical reactions are taking things apart. But if you look at all of the reactions happening in a cell, we call that metabolism. There are two different kinds. The first kind of metabolism is putting things together, building things. Those are anabolic reactions, anabolic metabolism. Think, for some of you maybe have heard of this before, anabolic steroids. Those are the kind you put into your body to help you build muscle. So those are specific enzymes that would cause things to join together. In this case, to build muscle cell. And then there's catabolic metabolism or catabolic reactions, which are the opposite of anabolic. They're breaking things apart. So when we talk about digestion, it's all about catabolic reactions. We want to take those polymers and break them down into monomers. There's a fellow from a show called Alone. I don't know if anybody's watched that on TV before, but it's drop you off in some remote location. Actually, a bunch of people get dropped off in a remote location far enough apart that they have no contact with each other. They have no contact with anybody, actually. And they film themselves, and it's whoever is the last person standing, whoever can survive the longest on their own with no human contact, no food. You get 10 tools you get to bring with you. You get to pick. Whoever lasts the longest wins a whole bunch of money. And you don't even know how many people are left and who's tapped out and who's still there. So this is a picture of a guy who basically starved. The show is really an exercise in starvation, and the people go crazy. It's kind of strange. But that fellow is definitely not on anabolic steroids. There's no building. His body is simply breaking down all the reserves that it has. Enzyme function. Just to throw some terminology at you. The active site of an enzyme is the part of the protein where little molecules will actually come and land. It's the part of the enzyme that's actually involved in the reaction. The substrate is the thing, the molecule that binds to the enzyme, either to be joined to another one if it's anabolic, or to be split up into smaller pieces if it's catabolic. So the active site is just sort of like the landing strip on the enzyme. Almost all enzymes have a lock and key model where they have a specific shape, so they will only allow certain molecules to bind. Enzymes are very specific. They don't join or break apart anything. It is only specific molecules. So it's like a lock and a key, right? You can't grab a key and go get into any lock, just like you can't grab some molecule and grab a single enzyme and think that you can break it down. Only one active site to match up with that one substrate. And then what a lot of enzymes do, which is kind of weird, I don't really have an analogy for this, is that when a substrate binds to that active site, to the landing strip, and they're the right shape and they match nicely, this lock and key model, quite often the active site actually changes its shape ever so slightly to make an even better fit, which makes the chemical reaction happen even better. So that would be like having a key that kind of sort of fits in the lock, and if you get it close enough, you can get it in the lock, and then the lock would actually change, and then it would work really well. Sort of a strange analogy that doesn't really work, I'm sorry, but that's what induced fit looks like. The enzyme will actually change shape a little bit to match again, just that one specific substrate. That doesn't mean you can throw some other random substrate at it and have it change its shape. It's still one lock, one key, but it changes its shape ever so slightly. There's a little picture showing that the substrate lands, it bonds, it actually changes its shape ever so slightly. This could be digestion, some catabolism. You can see that enzyme's job was to break that substrate into smaller pieces, maybe from a polymer into smaller chunks or into monomers. So there's a synthesis reaction where you're putting things together. You can see that there are some multiple substrates right here that fit nicely into the active site on the enzyme, and this enzyme's job is to join them together. Remember, that's an anabolic reaction. Could also call it synthesis, where we put them together and hey, look at that, multiple things turned into one product. So that's how a synthesis or anabolic reaction could happen. And the reverse of that, a degradation reaction, which is catabolism, breaking things down, might look like this, where you've got a substrate. You can see that its shape matches the active site on the enzyme very nicely. It's like a lock and a key. So it binds in there, and then the job of the enzyme is actually to break it apart, to split the thing into smaller pieces. So that's a degradation or catabolic reaction. And you can see then how the sort of lock and key model works between the substrate and the active site on the enzymes. When you're looking at enzymes, quite often their name will give you an idea. Don't memorize this chart, by the way. But their name will give you an idea about what substrate they're either putting together or breaking apart. So, for example, lipids are broken down by lipase, and maltose is broken down by maltase, and lactose is broken down by lactase. So for a lot of these things, when you see that ending ASE, that tells you that it's an enzyme, and then the sort of name in front of it is the substrate that it's working with. So how do enzymes speed up reactions? We know that pretty much every reaction in your body needs an enzyme to go faster. It turns out they lower something called the activation energy. So every chemical reaction, you have to put energy in to get the reaction to actually get going. And what enzymes do is they require less energy to go in to get the reaction going. So here's a chart. It shows you this solid line. You can see this big lump here. There's this huge amount of energy that would be required to actually get the reaction going. The reaction starts once you start to see this downward slope. So the activation energy without the enzyme is very high, but with the enzyme's help, you can see that we've lowered that energy required to get it going. In fact, it only took a little bit of extra energy, boom, and then the reaction goes, and we get the products. So lower the amount of energy, lower the amount of joules required to get a reaction going, and you can get more faster reactions happening. So there you go. Big, bold, colorful letters. Enzymes lower the activation energy, more reactions, faster reactions. Enzymes are impacted by lots of different things. We're going to spend a few minutes on that to finish off the lesson. Number one is the concentration of the substrate and the enzyme. So the more stuff you put in there, the easier it is for those substrates to bind to the enzymes. You'd have faster reactions. Or the more enzymes you have, you'd have more reactions. So the concentration of both the enzyme and the thing the enzyme is using will affect the reaction rate. If either of the substrate or the enzyme concentration go up, then the reaction rate goes up. There you go. More reactants, more substrate means more reactions can occur. RxNS is just short for reactions. Another really important one is temperature. So in our body, almost all of the enzymes worked at an optimal temperature of 35 to 40 degrees Celsius. when we cool things off too much, enzymes slow down. And when we heat them up too much, they slow down. And if you heat them up like way too much, then they actually denature, which you'll remember that term from the last video. And that means the three-dimensional shape changes, and we typically can't fix that. So all enzymes have optimal temperatures to work in. In fact, your internal temperature would be different than the temperature in your hands. So that means digestive enzymes are probably functioning more at 37, 38 degrees Celsius, whereas the enzymes that are allowing your hands to move, they're functioning at a slightly lower temperature. So they'd work just fine maybe at 35 or 30 degrees or even colder than that. Although you do know that when you get cold, at least for me, my hands are always cold, and the colder they get, the slower they move, because those chemical reactions slow down. So you have to find a sweet spot for temperature. You can't say that low temperatures are slow and hotter is faster. You have to sort of nail in the right temperature. And the third one is pH. There's a sweet spot for temperature. There is also a sweet spot for pH for all enzymes. Most of them work in the range of 6 to 8 for pH. That's neutral. Speaking specifically about digestive enzymes, pretty much the entire digestive system is a pH between 6 and 8, other than your stomach. So remember, your stomach is very acidic. It has pepsin, which is a special enzyme because it works great where the pH is really low. But if you took pepsin and you moved it into your small intestine where the pH goes back up to about 8, it wouldn't work very well. In fact, it probably wouldn't work at all. And if you took some of the enzymes from your small intestine and put them in your stomach where it's super acidic, it would denature those enzymes and they wouldn't work at all there either. So they need their specific happy place for temperature, and all enzymes need their specific happy place for pH. Inhibitors are things that can actually bind to enzymes to slow them down. So a competitive inhibitor is something that binds to the active site of an enzyme. It sort of fills in the active site so that the regular substrate can't bind. So as an analogy, that would be like the lock and key model. That would be like if somebody had a key that didn't quite work in the lock, but it fit, and they could jam it in there. And then you show up with your key, and your key doesn't fit because there's something already in the lock. So a competitive inhibitor fills up the active site, and then the substrate doesn't have any room to actually get in there and bind like it should. I'll show you a picture of that on the next slide. There's also some other inhibitors that are interesting called non-competitive inhibitors. So they bind to a different spot on the enzyme. So we call it the allosteric site. So this term here, allosteric site, which is not the active site. It's kind of like a back door. And when they bind to the allosteric site somewhere else on the enzyme, it actually causes the active site to change shape. And then we lose that lock and key fit, and the enzyme doesn't work anymore. So the substrate won't fit because the sort of lock has changed shape. So here's a picture to show you. This first one here is your competitive inhibitor. You can see the blue thing here is the competitive inhibitor. It has sort of put itself in the active site. It's sitting in the active site, so this substrate, which usually would fit in there beautifully, no longer fits because something's jammed in there already. It can't bind. That enzyme is going to be non-functional. And then your non-competitive inhibitor binds at some other spot, and you'll notice here that it's changed the shape of the active site. We've sort of curled the edges in, for example, and now the substrate can't fit in. It's ruined that lock and key fit. Just to give you an example, cyanide. You've probably heard of that before. It's a poison. It's a non-competitive inhibitor that binds to a specific enzyme that's used in ATP production. So it binds to some random allosteric spot, not the active site, but some other spot on an enzyme that is responsible for making ATP, and it deforms the active site, and your body can't make ATP anymore. Can potentially cause death. And another one is penicillin. It's a non-competitive inhibitor that binds to an enzyme that bacteria use to make their cell wall. So it binds to the allosteric site, which changes the shape of the active site, and it makes that enzyme non-functional. Bacteria can't build their cell walls properly, and the cell doesn't survive. So that's how penicillin, that's a good one. Cyanide is not, that's a poison. But penicillin works by non-competitive inhibition on bacterial cell wall enzymes, which is kind of cool. Feedback inhibition, I'll just touch on this briefly. We get into it a lot in the reproduction unit. But what's kind of cool is enzymes are cranking things out for our body. They're breaking things down or building things up. But sometimes we need them to sort of slow down once they've broken down enough stuff or built up enough stuff. So a lot of times what happens is the product of the reaction can sometimes bind back onto the enzyme to slow things down, just to keep things in check so that your body is not constantly breaking stuff down or constantly building things up. You could think about constantly breaking things down would potentially be bad, to have that catabolism happening all the time. So this feedback inhibition says that the products of reactions can actually go back and turn off some enzymes and slow things down, just to keep everything going moderately. But again, we'll chat about that in more detail. The last things that can affect enzyme activity are cofactors. So these are cool. It's like non-competitive inhibition. So these fit into the allosteric site. But instead of making that turn the enzyme off, instead of making that change the active site in a bad way, it actually changes the active site to fit the substrate even better. So it's the exact reverse of non-competitive inhibition. Cofactors bind to the allosteric site and they improve the fit between the substrate in the active site. And examples of that, organic just means containing carbon. Vitamins. So when you're talking about different vitamins like vitamin D, vitamin C, those are actually going to bind to enzymes in your body, specific enzymes, to help them function better. Heme and flavin are other ones. I don't know if you've heard of those, but vitamins are a big one. And then there are non-organic cofactors, which means they don't contain carbon. And these are things you'd get. Sometimes we call these vitamins, but they're in multivitamins, but they're not vitamins themselves. They're metal ions. So things like magnesium, copper, manganese, all the different elements on the periodic table that we need in very small amounts, because they help change the active site of certain enzymes to fit the substrate better. So there's enzymes. If you have any questions about this, please email me or join me in the next Zoom session. I hope to chat with you very soon.